We have another trial called the PROMISE study, which is screening higher-risk individuals who are at increased risks of developing MGUS and smoldering multiple myeloma, either because they have a family member or are a first-degree family member of someone who has multiple myeloma, or because they're of African American descent. Both of these populations in the previous research have been shown to have increased risks of precursor disease. When we look at the way that myeloma develops, we need to start at the cellular level and go all the way back to the beginning of MGUS. When does MGUS develop? So what we've used is mass spectrometry to look at the very fine level at the bone marrow and in the circulating blood to see how MGUS patients develop from the onset. So starting at even like that 1% plasma cells in the bone marrow. And what we found from the PROMISE study, and this is work done by Habib from our lab, we found that there's a novel condition called monoclonal gammopathy of undetermined potential. And this is patients who have an M spike that can't be detected in the serum using standard techniques from the clinic that has to be done using mass spectrometry. And so with the use of MGIP, we're hoping that we can come even closer to the very origin of the development of MGUS and therefore better understand both the biology of how the disease develops in the first place, which would then hopefully help us to prevent the progression to overt myeloma, but then also how we can prevent progression within the precursor disease states as well.